Okay, great. I just wanted to start out by thanking Paper Trails for giving me the opportunity to present my research. My name is Twishi, and I'm a current student at the University of Colorado Boulder studying biochemistry. And today I'm excited to share the summer project I've been working on at the University of Colorado Anschutz as a part of the Gates Institute summer internship program in Dr. Chelsea Magan's lab. Since this was a summer project, these are mostly preliminary results, but my research focused on using hydrogel-embedded precision-cut lung slices, or PCLS, as a promising ex vivo model for studying idiopathic pulmonary fibrosis. Idiopathic pulmonary fibrosis, otherwise known as IPF, is a severe lung disease associated with high morbidity, characterized by repetitive alveolar injury of unknown cause. Environmental factors and aging-related changes can trigger epigenetic reprogramming, leading to abnormal activation of the epithelial cells. These activated epithelial cells secrete a large number of cytokines, such as TGF-beta, which promote fibroblast migration and proliferation, and further induce fibroblasts to differentiate into myofibroblasts. And these myofibroblasts then secrete large amounts of extracellular matrix, leading to excessive deposition. And the ECM normally acts as a supportive framework for cells, but the overproduction causes tissue stiffening, a process otherwise known as fibrosis. And this tissue stiffening, in turn, makes it increasingly difficult for patients to breathe. Additionally, the repetitive injury leads to the death of alveolar type 1 cells, otherwise known as AT1 cells. And these alveolar type 2 cells attempt to compensate by differentiating into AT1 cells, but as the injury persists, AT1 cells continue to die, and the pool of AT2 cells becomes depleted. And then this creates a vicious positive feedback loop, exacerbating the disease progression. Despite the severity of IPF, it remains severely under-researched. Currently, only 2 FDA drugs are available to slow its progression, and there are no definitive therapeutic interventions. This is where PCLS comes in and offers a promising alternative. PCLS is an emerging ex vivo model that effectively models chronic lung diseases. I like to think of it as a microcosm of the lung, as it involves taking a small section of lung tissue that retains the characteristics of the entire lung architecture. This method allows researchers to produce a large number of replicates for experimental studies without relying on extensive animal models. My project specifically focused on exploring its potential for modeling IPF. Specifically, we aim to determine if PCLS samples could be maintained in culture over an extended period of time, and whether the samples could exhibit fibrotic-like characteristics throughout the experiment. This aspect is particularly novel because PCLS models have rarely been studied beyond a one-week time frame in the context of pulmonary fibrosis. To engineer this PCLS-based model, we began by filling mouse lungs with agarose via the trachea and then cutting them into thin slices, which were about 500 micron thick slices using a vibratome. These slices were then uniformly shaped with a 4-millimeter biopsy punch and then incubated overnight in complete culture media. The next day, the PCLS punches were embedded in a custom silicone mold with 8-millimeter diameter wells, and we cast a base layer of the hydrogel and polymerized it to covalently bind to the top and bottom layers and placed the PCLS punch in the center. A top hydrogel was then added and polymerized to create a solid structure. The hydrogels were either a soft or a stiff norborine conjugated 8-arm 10K PEG macromer, which we were able to functionalize to 93.5%. And we also added an MMP9 crosslinker and peptides to promote cellular adhesion and interaction. These hydrogels were designed to mimic the different stiffness levels found in healthy versus fibrotic tissue. If you take a look at the rheology results, we were able to tailor the stiff hydrogels to match fibrotic lung tissue stiffness, which falls over 10 kilopascals based on prior studies. And we were also able to tailor healthy lung tissue to fall between 1 to 5 kilopascals. The embedded PCLS punches were then cultured in free-floating standard media until analysis. 4 days post-embedding, half the samples were exposed to a fibrosis cocktail, which I will now refer to as FC, that could create fibrosis-like activity in the sample. The fibrosis cocktail consisted of T oligo, which creates DNA damage, TGF-beta, which is a key activator of fibroblasts, and platelet-derived growth factor, which drives the production of the extracellular matrix. The other half of the samples were exposed to a vehicle control, which was essentially the negative control for the experiment. Media was then replenished every 48 hours, and FC or VC was administered every 4 days. And on days 1, 8, and 15, assays for cell viability, metabolic activity, and specific gene expression were performed. Looking at the lab data images that represent the sample pool, you can see the viability remained high throughout the 2-week experiment. And the day 1 samples before the FC or the VC administration was a healthy tissue, and the day 15 VC sample and day 15 FC sample both indicate that the tissue maintained its structure, with cell viability slightly decreasing over time, particularly in the FC samples. Notably, the cell death observed at the tissue edges aligned with areas induced with trauma, and it was expected to see more cell death in the fibrotic samples since the cells are hypoxic. And this trend was consistent across both the stiff and the soft experiments, although the images shown here are just from a soft hydrogel experiment. The Presta Blue metabolic assay demonstrated an increase in metabolic activity from week 1 to week 2, and as expected, FC samples consistently showed higher metabolic activity than BC samples, because that's what is technically observed in vivo, and this occurred in both the soft and the stiff experiments. The PCR heat map from the stiff hydrogel experiment revealed gene expression patterns that are highly indicative of fibrotic lung tissue. We specifically observed an upregulation of the key fibroblast markers such as FN1, COL1A1, CTGF, LTBP2, and CTHRC1, alongside the transitional epithelial cell markers, hypoxic cell markers, and the AT1 markers, which are all highlighted in blue. And then conversely, there was also a notable downregulation of the AT2 markers, specifically surfactant protein C, which was critical for maintaining alveolar epithelial function. And as I mentioned earlier, during the first week of the experiment, we observed a significant upregulation of the fibrotic genes, alongside with the decrease of SFTPC expression, indicated by the reprogramming of the cells. But this distinct profile underscores sort of the effectiveness of our PCLS model in replicating key aspects of pulmonary fibrosis. But the results in week 2 were a bit more variable. Based on the advice from my PI, I plan to extend the experimental timeline to observe if a more consistent trend emerges. And the pronounced week 1 results likely reflect an acute fibrotic response as the samples reacted to the fibrosis cocktail. But then by week 2, as the samples attempted to reach a state of homeostasis, the variability in the results increased. And this extension could help clarify whether the model can maintain fibrotic states over a longer period of time. And in conclusion, the results from our study indicated that this PCLS model provides a physiologically relevant 3D microenvironment for studying pulmonary fibrosis. It successfully recapitulated the complexity and cellular and extracellular components observed in fibrotic tissue for the most part. And additionally, this model offers a pretty great option for sex and patient-specific studies to take place to study fibrotic initiation and disease pathogenesis. And moving forward, several areas require further exploration. First, we need to conduct a stiffening experiment that models the transition from healthy to fibrotic tissue. This can be achieved by stiffening a soft hydrogel with ruthenium and SPS, and that would be a great way of achieving that stiffness. And another crucial step would be increasing the number of replicates, because we need to see more consistent trends across the experiments and achieve statistical significance in the results. And furthermore, introducing the 2 known FDA drugs, nintedanib and pirfenidone, could help in determining if the induced fibrosis in the tissue in the model can be mitigated. Lastly, it's essential to validate this model using human lung lobes and explore its utility in studying other lung diseases. The most important takeaway from this research is the great potential PCLS has in preserving the lung tissue architecture and reducing unnecessary animal testing. Our lab's hydrogel formulation allowed these samples to be maintained in culture for up to 6 weeks, offering ample time for high throughput studies, which is a pretty exciting prospect for future research. Lastly, I would like to acknowledge the Magan Lab for their support and guidance through this project, especially Dr. Chelsea Magan and Alicia Tannberger. And additionally, I'd like to thank the Gates Institute and the Gates Summer Internship Program for providing funding for my work this summer, the donors of which are listed below the picture. And that concludes my presentation.